Okay, so we're going to pick up where we left off. We just finished up talking about vascular plants as our examples of photoautotrophs and how they acquire nutrients and spread them throughout their plant bodies. And now we're going to talk about chemoheterotrophs using fungi and animal as our examples for how they acquire nutrients as well. So chemoheterotrophs in general, they can be broken down into a lot of different ways, but one way they can be broken down in how they acquire nutrients is as absorptive heterotrophs versus ingestive heterotrophs. So absorptive heterotrophs include examples like fungi and the prokaryotes. So remember, prokaryotes are things like bacteria and archaea, and those can absorb small molecules that are present from the environment and then move them across cell membranes because they're really small molecules into the interior of the cell. Or fungi can actually secrete enzymes out into the environment that digest and break stuff down outside of their bodies. And then once those molecules are broken down small enough, they can be moved across their cell membranes into the body of the fungus. So this is an example of an absorptive heterotroph. In contrast to ingestive heterotrophs, and only eukaryotes are capable of ingestive heterotrophy. An ingestive heterotrophy, all it is basically taking things into your body and then breaking them down. So in some cases, unicellular eukaryotes like protists can basically do phagocytosis, so bring a big chunk of food in and break it down once it's inside of their cell. Or animals like us are extracellular ingestive heterotrophs, where basically you bring a piece of food into your digestive tract. So technically that's sort of like inside of your body, but it's still outside of your individual cells until it's broken down and digested and then eventually absorbed across cell membranes and gets into sort of like your cells and your bloodstream and things like that. So just to sort of make this a little bit easier to remember, just remember that absorptive heterotrophs break things down, then take them in across their cell membranes. And absorptive starts with A, B, and B comes first in break. So absorptive, break, then take. And then ingestive heterotrophs take things in and then break them down. So we're going to look at fungi as an example of an absorptive heterotroph. Remember, fungi are eukaryotes located here on our sort of larger phylogenetic tree close to this branch with the animals. So fungi, hopefully you can recall, typically when you think of a fungus, you probably think of a mushroom, but spend most of their life underground as this mycelium life stage, which is basically sort of a lot of little threaded roots and hairs that run through soil or whatever substrate the fungus is growing in. And this is the feeding life stage. So it's a filamentous structure that has a really, really high surface area to volume ratio. And that's not only to aid with absorption of nutrients from the environment, but it'll also secrete enzymes into the environment that break stuff down in the environment. So it aids also in digesting nutrients that are out there in the environment and breaking them down before they're absorbed. So this mycelium aids in both digestion or breaking down of nutrients in the environment and absorption of those nutrients once they're broken down from the environment. And just to give you an idea of how high the surface area to volume ratio is in mycelium in the soil, it has the highest surface area to volume ratio of any organism known to man. And it's estimated that a cubic inch of soil can have as much as 1 mile of mycelium from one fungus threaded through it when it's really sort of densely packed. So incredibly high surface area to volume ratios. And so this should fall in line with that sort of overarching themes that we talked about related to structure and function and trade-offs. So structure and function, meaning that because these fungi are involved in breaking down and absorbing nutrients from the environment, it makes sense that they'll have a really high surface area to volume ratio so that their structure matches that function of absorbing nutrients from the environment. But this is also a big trade-off. As with anything, when you have a really high surface area to volume ratio, you tend to lose water really quickly, and most fungi lose water to their environment really easily. And this is part of the reason that you tend to only find fungi in moist, wet places and not really dry places, is because they just dry out way too much and can't survive because they have this really high surface area to volume ratio in their mycelial life stage. So given that, please answer this question. Really all this question is asking is comparing this type of fungi, hyphae. So think of a single like thread of mycelium, if it were sort of thick in a big cylinder like this, or if you have the same volume of fungi broken down into a bunch of little thinner hyphae. Which of these is going to be more or less efficient, given that they have an equal volume, more or less? Going once, going twice, 3 times. So 93% are saying A, the thicker fungus or the thicker one is going to be less efficient than this one with lots of thin fungi that have the same overall volume. And that is the correct answer. And that's simply because this sort of layout has more surface area per unit volume for digesting and secreting enzymes and breaking stuff down, but also absorbing nutrients from the environment than this one. So higher surface area to volume ratio here. Higher surface area to volume ratios are typically better at absorbing nutrients from the environment or, secreting enzymes into the environment like we might see. So here's another sort of review slide, this idea that structure reflects function. Which of the following is false or does not support this idea? This is going over stuff from many past lectures as well. Going once, going twice, 3 times, and sold. 86% are saying C. C is the false statement. When you're smaller, you have a higher surface area to volume ratio. So a smaller single-celled prokaryote is going to be a little bit more efficient in taking in nutrients per unit volume because it'll have a higher surface area to volume ratio than a larger eukaryotic cell. Any questions about any of these other choices? So fungi as absorptive heterotrophs need to get carbon atoms, carbon molecules, and energy and other essential nutrients. And they can do this in 3 sort of ways. One is they can get high energy carbon molecules from dead things. When fungi consume dead things like this dead tree or this dead log here, they are considered to be what's called a saprophyte. And that just means that they're living off of dead stuff and getting their nutrients from something that died previously. They can also live as mutualists, like we learned about with the mycorrhizal fungi that associate with plant roots, where they give nutrients from the soil to the plant roots, and in exchange, they get high energy carbon molecules, aka sugars, from the plant. And they can also live as parasites. So a parasite is a symbiotic relationship where one organism benefits and the other organism at the cost of the other organism. So it's a detriment to the other organism. This is an example of one parasitic fungus. There's many different kinds, but this is called corn smut. And you can see it's a fungus that grows on corn and takes over its nutrients while it's still alive, or takes nutrients from it while it's still alive. And you can actually buy this to eat. It's considered a delicacy in some cultures. So this is huetlacoche, and I've had it before. It's actually pretty good, even though it looks kind of gross. So in all of these cases, whether it's a saprophyte, a mutualist, or a parasite, these fungi are feeding on complex carbon molecules that store a lot of energy. So in this case, they're often eating cellulose and lignin, which are these sort of complex carbohydrates or polysaccharides. Same deal with the huetlacoche. It's feeding on starches, which are another example of a bunch of glucose molecules stuck together to make these large, complex polysaccharides or carbohydrates. And so it's a big challenge for fungi to eat these sort of big molecules because they don't have a mouth and they don't have a digestive tract. They also have cell walls that prevent them from doing phagocytosis and breaking in sort of larger chunks of that stuff. So they need a different way to break things down. And the way they do that is by basically secreting digestive enzymes. And the reason they need to secrete these digestive enzymes is that these larger molecules, like cellulose, like lignin, these polysaccharides, will not move across the cell membrane naturally. So here we're just looking at a phospholipid bilayer or a cell membrane. And here we're looking at a sort of scale of permeability for different types of molecules. And so these molecules up at the top, like O2, CO2, N, it turns out they can move very freely and very easily across the cell membrane without much challenge. So these move across very easily, small nonpolar molecules. Whereas if you look at things like H2O and glycerol, these are still small molecules, but they don't have a charge. They only have like a little bit of what's called polarity. So maybe you remember learning about H2O. It's sort of a polar molecule where it's got a little bit of a negativeness to it on one side and a little bit of a positive to it on the other side. And these still move relatively freely, but not as easily. And then you get into larger molecules like glucose and sucrose, which can move across, but most of them bounce off and don't penetrate a cell membrane. Until lastly, you get to things like ions that just cannot break through a phospholipid bilayer or a cell membrane at all. And so if you put these molecules like cellulose, like lignin, like starch that fungi are feeding on to get carbon and energy, most of them fall into this area of being like an ion that they just basically bounce off because they're just so large that they cannot move across this cell membrane. And so what that means is fungi need to digest those molecules and break them down into smaller molecules first before they can move them across their cell membrane. So food is digested by the hyphae that secrete these digestive enzymes outside of the hyphae and break down things like cellulose, things like lignin from these polymers into the individual glucose monomers that make them up. And then those glucose monomers can more easily move across the cell membranes and get into the actual fungal cells so they can take on some of those higher energy molecules and get nutrients. And so this is really important for larger scale carbon cycling. As it turns out, fungi are some of the only organisms that are able to break down cellulose and lignin and the heavy stuff that makes up trees. And if we didn't have fungi around, likely many, many trees would be building up trees and leaves and all this stuff that's made out of cellulose and lignin on the ground so that we'd be, buried in meters and meters and hundreds of feet of trees that never broke down. So the fungi really help break that stuff down and move carbon around through carbon cycling. And so just to sort of put this in a more visual, display this in a more visual way, you can think about a carbon atom in red here that's trapped in the atmosphere. And as a part of photosynthesis, those carbon atoms are built up into the biomass of trees. Some of that carbon is brought back into the atmosphere when plants do cellular respiration, but much of it is trapped up in the trunk. And when that tree dies, all of those carbon atoms are trapped in that dead tree as cellulose and lignin and these complex molecules that are difficult to break down. And then fungi come around And they have these specialized enzymes that are pretty much unique to fungi and maybe a few species of prokaryotes that are able to break down and break apart these bonds that make up cellulose and lignin and take that carbon that's trapped here in the trees and move it into their own biomass. And the fungi in turn, so some of that carbon once again gets turned into the biomass or converted into the biomass of the fungi, but those fungi in turn will also do cellular respiration and return some of that carbon back to the atmosphere too. And so this is essentially carbon cycling, where these carbon atoms are moving from the air to a tree, from a tree to a mushroom, from a mushroom or fungi back into the atmosphere. And once again, fungi are unique in their ability to break down cellulose, lignin, and these really tough to break down molecules that exist in trees and plants. So given that, which of the following statements is true? Going once. Going twice. 3 times. Sold 44% are saying D, 26% are saying E. The correct answer is in fact D. So fungi are not ingestive, they are absorptive heterotrophs. They break things down and then take them in, as opposed to ingestive heterotrophs, which take things in and then break them down. And we'll talk more about that in the next couple of slides. Animals are ingestive heterotrophs. Fungi use biosynthesis to incorporate carbon into their biomass, not cellular respiration. Cellular respiration is basically going to turn carbon atoms that's in their biomass into CO2 and put it back out into the atmosphere. Saprophytic fungi break down cellulose from dead things. So saprophytic is dead stuff. Parasitic is going to break down from living things. Plants or animals. Any questions about any of those choices? All right, let's try this one. Going once. Going twice. 3 times. And sold. 20% are saying A, 69% are saying D. Nice work. 20%. You are correct. You remembered that nitrogen is not a part of polysaccharides. Polysaccharides are just made up of carbons, hydrogens, and oxygens. And so this is a bit of a quote unquote trick question. Although nitrogen is an important macromolecule that basically all organisms need to survive, nitrogen is really only a part of amino acids and nucleotides. So you can see this nitrogenous base here. You can see this amino group that's going to be on all amino acids and nucleotides, but nitrogen is not part of polysaccharides. Any questions about this one? Okay, so very quickly, we've moved through fungi as example of our absorptive chemoheterotroph, and now we're going to talk about animals as our example of ingestive chemoheterotrophs. So everything you're seeing on this image here, whether you're talking about an ant, an anemone, a jellyfish, a snail, a worm, a crayfish, a hamster, everything on here is an example of an animal, I guess, except for the carrot in this hamster's mouth. Animals, hopefully you all know by this point, they are all eukaryotes. They don't have cell walls. They are all multicellular. They are all chemoheterotrophic. They all have some life stage that is motile. In other words, it can move under its own power. It includes invertebrates and vertebrates, so things with backbones and without backbones. And when we talk about nutrient acquisition in animals, as we mentioned before, animals are what are known as ingestive heterotrophs. They take things in to their digestive tracts and then break them down once they're sort of inside their bodies. And whether you look at an earthworm, a grasshopper, a human, or a dog, all of these things are animals, and all of these things have the same sort of major setup or layout of their guts or digestive tracts, where they have what's called a foregut, like you see in green in each of these animals, a midgut, like you see highlighted in sort of reddish pinkish in each of these animals, and a hindgut, like you see highlighted in yellow in each of these animals. And the foregut typically consists of a mouth, an esophagus, and a stomach involved in sort of taking things in and breaking them down. The midgut typically involves the small intestine that's involved in absorption of nutrients from the sort of digestive tract across cell membranes and eventually into the bloodstream so it can be transported throughout the body. And then lastly, the hindgut, which typically consists of a large intestine, rectum, and anus, which is involved in, of course, elimination. So just to look at this another way in how animals acquire nutrients, whether you're a worm, a dog, a human, basically there's a couple major steps. The first one being ingestion, which is the act of eating or feeding or bringing something in. So ingestion is bringing in packets of food. And typically, in the case of humans, this involves the mouth and esophagus, which is the tube leading down into the stomach, where the next step happens, which is, of course, digestion, where food is broken down into molecules small enough to be absorbed across cell membranes. So this is not just like grinding it up and making it into little pieces, but it's also chemically sort of dissolving it and breaking it down, not just into smaller pieces, but into its smaller chemical constituents so that it can become a molecule that's small enough to be absorbed across a cell membrane. Next is absorption. So this is when cells take up those small molecules now across their cell membranes. And this happens almost exclusively across the walls of the small intestine, maybe a tiny bit in the large intestine, but the large intestine for the most part is involved in elimination. So this is when undigested materials are basically packed together, have the last little bit of water removed from them, and then are removed from the body through the large intestine, rectum, and anus. So let's look at ingestion in a little bit more detail. Is it gonna let me switch slides? There we go. So there's several different strategies that animals have evolved for ingestion. So remember, ingestion is just taking it from the outside and bringing it into the digestive tract. One is called suspension or filter feeding, where you collect organic matter that's suspended in water and then move it into the gut. Baleen whales are a great example of these suspension or filter feeders. They have this thing called a baleen that basically looks like a comb with a whole bunch of hairs at the bottom. And so they'll find a bolus of like a bunch of little krill, which are tiny little shrimp, and they'll close their mouth around it. And the krill will move through this comb and get caught in all those hairs. And so they're like filtering the food out of the water. So this is a suspension or filter feeder. You can also have substrate feeding, like you see in the hungry caterpillar that basically moves through its substrate and eats it as it goes. So this is what caterpillars do. This is what worms do. And they basically leave a little trail behind them as they move through that substrate and sort of take it in. You can have fluid feeders. So mosquitoes are a great example of that. They obviously stick this long sort of proboscis type string thing into your circulatory system and your capillaries and suck the fluid out, in this case, blood. There's other fluid feeders that can feed on nectar. So for example, butterflies have a long straw that they suck nectar out of flowers with. You can also have other aphids and things like that that'll basically tap into a plant phloem and suck the phloem and the sugars out of a plant. So fluid feeding is another example. And then there's, of course, what we're most familiar with, which is how we eat, which is called bulk feeding, where you ingest large pieces of food and kind of chomp them down and then break them down further once they get into your digestive tract. So let's talk about bulk feeding in a little bit more detail and some of the key adaptations that are involved in bulk feeding. One of the most important or two of the most important of which are jaws and teeth. So here we're looking at a really big phylogenetic tree. And here you can see like our most recent jawless ancestors called the jawless fish, like hagfish and lampreys. So these are examples of jawless fish here, right? And you can see their mouths, they kind of look like, I don't know, like the sandworms from Dune or something like that, where they don't really have any jaws to speak. They just kind of suction cup on with these little teeth and then suck liquid and nutrients from the thing that they're preying upon. And these are common parasitic types of fish. And so somewhere jaws evolved in all of these other major groups, including mammals, sharks and rays, frogs and salamanders. All the sort of animals that we typically think of are examples of jawed animals, or I guess vertebrates that we typically think of are jawed animals. And the general consensus is that jaws evolved from gill arches. And so this is a fish. Here's its skull, or a lamprey sort of looking ancestor, and it has this cartilage all along the gills. And you can see its gills right here. There's cartilage that holds this up. And eventually, over time, that cartilage towards the front of the mouth became modified to form jaws that at first had no teeth, and then eventually teeth developed in there. And this, of course, was super helpful with biting off huge chunks of food and kind of processing and eating more food, like we see in things like sharks, which have these incredibly nasty jaws, or we've actually, in eels, they have two sets of jaws, which is kind of crazy. So the first set of gill arches evolved to this first set of teeth, and then within that, they have a second set of gill arches that has a second set of teeth inside their throat. And so have you ever seen the movie Aliens, when it like opens up its mouth and then the second mouth shoots out and grabs the prey? That's what eels have evolved, the same deal. They can open up their mouth and that second set of jaws grabs the fish and pulls it into its mouth, and then it clamps down on it and eats it. It's wild what evolution can come up with. So along with jaws, there's also, of course, the evolution of teeth. And if you look at how teeth have evolved in different types of organisms, depending on their primary diets and eating habits, you can see that the structure of the teeth is highly correlated or related to the function or the type of diet that those animals have. So if you look at, for example, herbivores and their jaw and tooth layout, you can see these different colors here: orange, blue, red, and green. Orange represents molars for grinding, premolars for shearing, and then incisors and canines for cutting and ripping and tearing. And so these have evolved a sort of layout in their jaws and teeth, perfect for kind of nibbling a little bit of grass and then passing it back to these molars and premolars for grinding it up into like a mash. Whereas if you look at carnivore teeth, they have really sort of large canines and incisors for just gripping, killing, and shredding apart pieces of meat and sort of gobbling down big chunks of meat. Or if you look at humans' teeth, we are omnivores. We eat both plants and animals, and we have sort of a combination of this ability to sort of rip stuff up, but also grind it up. So we can do a little bit of both. And so, as I said before, the structure of different teeth relates to their function. teeth and jaws relates to their function so that they suit the diets of these different organisms that they've evolved in. So food gets ingested. The next step is, of course, digestion. So once it's ingested or brought into the body, it goes into the stomach to be broken down chemically. When we look at the types of digestion, you can have in some animals what's called extracellular digestion, which is the breakdown of food outside of the cells and compartments continuous with the animal's body. So this is basically what's happening in your stomach. That breakdown is happening outside of the cells. And you can have this broken down into two types of extracellular digestion. You can have extracellular digestion in what's called an incomplete digestive tract, or you can have it in a complete digestive tract. The main distinction between an incomplete versus complete digestive tract is whether it has one hole or two holes. So incomplete, there is just one single opening for food to go in. And then once it's sort of broken down and absorbed, the waste that's left, guess what? It's coming out that same hole. So this is an example of an incomplete digestive tract. And we see this in jellyfish and anemones. So the in hole is also the out hole. And once again, that's in contrast to what we typically think of, because it's what we humans have, a complete digestive tract that has a mouth and an anus. So an opening at one end and an opening at the other end, so that food comes in one side and comes out the other as waste. So in the stomach is where digestion happens. Food is chemically broken down. Once it's chemically broken down into smaller chemical constituents, then it moves into the absorptive portions of the digestive tract, which typically consists of a small intestine, which is both incredibly sort of twisting, winding, and long, but also is lined with all of these little fingers and villi. And as you can imagine, being really twisted and curvy and long, and also having all of these little villi or fingers surrounding the interior of the small intestine helps to, of course, increase surface area to volume ratios to really enhance the efficiency with which the small intestine absorbs nutrients from the food that's been broken down. So once again, structure is related to function. This long length and lots of fingers gives it a really high surface area to volume ratio for nutrient absorption in the small intestine. Right. Oops, you didn't see that. So I know I moved through all that crap very, very, very quickly, but I also know that this is stuff that I'm guessing most of you are pretty like intuitively familiar with, so I didn't want to spend too much time beating a dead horse, as they say. So go ahead and click in on this question, please. All right. Going once. Going twice. 3 times. And souls. 80% are saying D. D is the true statement. Fungi are absorptive autotrophs. That's false. They are heterotrophs. Most fungi have a high surface area volume ratio to decrease rates of water loss. No, it's to increase nutrient absorption. And it really doesn't help with maintaining osmotic balance because they lose lots of water. Saprophytic fungi get most of their carbon from a mutualistic relationship. Nope, saprophytic feed on dead plants or animals. So this is describing mycorrhizal fungi, not saprophytic fungi. Baleen whales are examples of ingestive heterotrophs. That is true. And for most animals, large nutrient molecules must be broken down before they can be ingested. So this is not true. Most molecules can be ingested, and then once they're ingested, they get brought down into the stomach, and that's where they get broken down and eventually absorbed in the small intestine. Any questions about any of this so far? All right, another question for y'all. Going once, going twice, 3 times. 21% are saying A, 28% are saying C, 11% are saying E. So talk to your neighbor. Let's try this one more time because it's kind of all over the damn place. All right, let's try it again. Ring in, please. Going one. Going twice. 3 times. So 23% are saying F, 48% are saying A, 22% are saying C. A is the correct answer. So yes, some protists are ingestive heterotrophs. So they can do what's called phagocytosis, where they're taking it into the cell and then breaking it down. So this is an example of intracellular ingestive heterotrophy. Insects have an incomplete digestive tract with their mouth and anus in separate locations. This is false, not only because they have a complete one, but also because an incomplete has the mouth and anus in the same location. The mouth and large intestine are locations where the vast majority of nutrient absorption occurs. That is false. The mouth is for ingestion and a tiny, tiny bit of digestion because there's enzymes that can break stuff down chemically in your saliva technically, but the vast majority of nutrient absorption is going on in your small intestine. Small intestine is where nutrient absorption happens. It's advantageous for locations of nutrient absorption to maintain a low surface area to volume ratio. No, that's false. They should have a high surface area to volume ratio so they can absorb nutrients much more efficiently. Have a high amount of surface area so they can move lots of nutrients very efficiently across their cell membranes. And then the four main parts of nutrient intake for animals are ingestion, digestion, absorption, and elimination, not inclusion or expulsion or excretion. So ingestion, taking it in, digestion, breaking it down chemically, absorption, moving it across the small intestine cell membranes into the bloodstream so it can be spread around. These are the individual sort of nutrient constituents, and then all the leftover stuff is eliminated. And so most of you are pretty, I think, intimately familiar with this process where you eat, you poop, and it's all sort of super simple. Like you've been doing it since you were less than a year old, right? But as it turns out, this process of ingestion, digestion, absorption, elimination, chemically speaking, is incredibly complicated. And there's this guy, his name is Wim de Volier. He's an artist and a chemist and a scientist who has these art installations that he calls his cloaca series. Cloaca is Latin for sewer. Essentially what he's done is he's built a series of sort of flasks and chemicals and stages where he takes food and he turns it into poop. So these are some of his cloaca machines. He has them all temperature controlled with very specific digestive enzymes and sort of mechanical processes. And you can go to these art installations and watch the machines run. They've been sold across the world for price tags ranging from anywhere to $200,000 to $1 million for these installations that essentially are just machines that create poop. Here's cloaca number 5. Here's super cloaca. And here's some of the products that come out of his cloaca machines. As you can see, they look just like poop. And at the end, many of these art, what's the word I'm looking for? The places that display the art, museums, that's the word for them. They will actually bag these up in little vacuum sealed things and sell pieces of turds for $15,000 a piece. And people will buy them because once again, it's art and it's poop that's created by a machine. So I think the point that I'm trying to drive home here is that this process that we take for granted is actually incredibly complicated. And this guy has dedicated a very significant amount of his life and time to figuring out every sort of step in this process. And it's complicated enough that other people are not able to replicate this. Like it's really, complicated to make poop, believe it or not. So yeah, how about that? Fun fact for the day. That is all. I will see y'all on Wednesday.